Добрый день, уважаемые участники нашей секции. Секция называется «Агротехнологии», и с нашей точки зрения это одна из ведущих секций форума, потому что форум, который проходит под эгидой Росатома, он объединяет не только вот эти новейшие технологии, которые включают в себя индустрию Росатома, но, как мы все понимаем, что без продовольственной безопасности и еды никакие новые технологии эффективными быть не могут. Поэтому в рамках этого форума мы сегодня с вами обсудим те новшества, которые есть в области аграрных технологий, те очень интересные наработки, которые могут быть имплементированы в странах с жарким климатом, то, что может быть сделано в совместной коллаборации между нашими учеными и учеными африканского континента. Секция будет вестись на двух языках, русском и английском. Вы можете свободно говорить как на русском, так и на английском. Мы поймем друг друга, мы можем задавать вопросы и отвечать на двух языках, русском и английском. Секция наша будет состоять из двух частей. Мы сейчас работаем до 12 часов, и после получасового перерыва вторая половина начнется в 12:30. Участников мы тоже ждем в этом зале. Дискуссия у нас будет свободная. Я вас призываю всех активно принимать участие, не стесняться задавать вопросы, потому что тематики очень интересные. Часть докладов будет в очном формате, часть докладов будет онлайн, но это все очень интересные тематики, которые, безусловно, обогатят нас с вами знаниями и добавят нам новых компетенций. Теперь разрешите передать слово второму модератору секции Сергею Николаевичу Иланскому для приветственного слова.
Здравствуйте, уважаемые участники, и очные, и заочные. Надеюсь, у всех удалось удачно подключиться, и нас хорошо слышно и видно. Сегодняшняя секция очень интересная. Раньше мы пытались делать доклады и приглашать людей, кто может сказать только про применение именно ядерных технологий. Но на этой конференции мы впервые приняли решение расширить круг обсуждаемых вопросов и обсудить многие вопросы, связанные с защитой растений, с технологиями возделывания, с технологиями дистанционного мониторинга, то есть все то, что составляет современную систему выращивания растений. И здесь очень интересно, прежде всего для африканских коллег, то, что в России налажены выращивание растений на больших площадях, работа в системе агрохолдингов, то, что очень востребовано сейчас в Африке. У них это очень слабо развито, и они очень интересуются этими направлениями. Ну и, наверное, я перейду плавно к своему докладу.
Коллеги, позвольте мне, now I want to say some words of greetings to those our colleagues who cannot understand Russian and who also must be involved to our discussion process. We are very glad to have opportunity to meet with all of you here in People's Friendship University of Russia, one of the best universities in Russian Federation and world over. Our widely and broad discussion will be related to the subjects of food security, of new technologies of agriculture, and all of you are welcome to participate in this discussion, to have his own opinion and to change this opinion with colleagues. Does not matter whether the colleagues are near you or they are on the other continent and they are speaking online. I wish all of us success. I wish all of us good ideas, exchanging of good ideas, and again, welcome in People's Friendship University of Russia. Thanks. So now we are going to first report, which will be given by Professor Yelansky Sergey, and the subject is cultivation and the protection of potato plants and tomato from fungal diseases in Uganda.
about agro-business in Uganda, a place of Ugandan potato production in African production, and problems of potato growers and tomato growers in Uganda. Next slide, please. Areas suitable for agriculture and especially for plant growing are not large in the world. The main areas situated in Europe, moderate climate part of Russia, in India, China, and North America. In Africa, very few regions are suitable and good suitable for plant growings. Next slide, please. But Uganda, situated in the altitude of 1,000 meters above sea level, near the freshwater Lake Victoria, and has several other rivers and lakes that can be used for water supply for plants. And you can see that in African map, Uganda has many green areas where the growing of plants is possible. But if you see to the right map of average dry yield for all crops, you can see that Uganda and almost all African territories are blue. So they produce very small amount of yield of different crops in comparison with Europe and North America. So Africa has a good possibility for growing of plant production. Next, please. Potato production in Africa is one of the main businesses in different countries. In Uganda, also potato is, I think, third crop after sugarcane, corn, and cassava. About 250,000 tons of potato are produced annually in Uganda. But the average yield is very small. 7 tons per hectare is considered as a very good yield for Uganda. For comparison, in Russia, average yield is about 26 tons per hectare, but in good companies, this amount is about 50 tons per hectare. In Europe, average yield also about 45 tons per hectare. What is the reasons of such small crop? Many different reasons. First of all is, next please. That is a typical landscape of potato growing area in South Uganda. You can see small fields of different farmers on the slopes of the hills. And the quality of soil is not good on these fields on the slopes. Next, please. It was done very interesting survey, which is introduced in the article of Harahagazwe et al. In 2018 year. 190 farmers from 19 sub-Saharan African countries were interviewed and asked about their vision of reasons of small crops and problems with growing of potato in their countries. And almost everywhere, people mentioned these reasons. First of all, low quality of seed material. development of the bacterium Ralstonia solanacearum in the soil. That is the largest problem in all tropical countries. Development of late blight. In Russia and in Europe, that is not a problem because everywhere good modern pesticides are used. But in African countries, people have no money to buy good pesticides, and their potatoes suffer from lead blight. Lack of fertilizers, and some people mention incorrect application of fertilizers, so people need good agricultural education to work on their fields. And the development of viruses is about 70%. So you can see that almost all these 5 reasons, the main reasons that they mention in their interviews, connected with problems with soil or problems with seed material. Next, please. And I'll tell a little about our survey that we have done together with Moscow Lomonosov State University and Makerere University of Kampala. We collected tubers and tomato fruits with symptoms of fusariosis or dry rot, extracted strains of fungi from these tubers or fruits, and estimated their species. We collected samples in 4 main potato productive areas of Uganda that is marked on the map. Next, please. We collected infected samples, isolated of arsenic cultures, tested their species, and made pathogenicity testing on potato tubers and tomato fruits. We did species identification using morphological criteria and sequences of specific regions of DNA that we compared with other sequences in GenBank using BLAST program, usually. Next one. So we isolated from potato tubers many different isolates of Fusarium. It is fungal genera Fusarium, very popular in potato, and in next slide I'll tell about Fusarium more. And other pathogenic species were Calitotrichum cacodes, Remotidimella destructiva, Helminthosporium solani, and Alternaria linaria on potato, and also Fusarium, Cladosporium cladosporioides, and Epicoccum on tomato fruits. Next one. The largest group was Fusarium, and we estimated the Fusarium isolates on their belonging to different species complexes. And we found that they belongs to 4 species complexes: Fusarium oxysporum, Fusarium solani, Fusarium sambucinum, Fusarium incarnatum equisiti. And it's interesting that one species of incarnatum equisiti, Fusarium flagelliforme in red frame, that is a new species for potato. It was never earlier found on potato. And in green frame, there are 3 strains from tomato. We think that that is a new species. We will make a work to write them as a new species, a new fungal species from Fusarium species complex incarnatum equisiti, because they have no similarity with any other Fusarium strains. Next one, please. Thank you. In this table, I introduced the fusarium species complexes on potato tubers in different countries based on different literature articles. So we can see that the most popular is a species complex incarnatum equisiti that can be found in almost every country of the world. Another one very popular species complex is Fusarium sambucinum. And in Russia, we have found more different species complexes, but maybe it's connected with the larger square of Russia. We collected in different regions and more isolates were tested. Next one. It's interesting that after testing of pathogenicity of our isolated strains, we found that almost all our Fusarium strains grow better on tomato slices of tomato fruits than on potato, and isolated strains isolated from potato tubers also grow better on tomato. It was interesting for us. Next one. And also all of tested strains can infect potato tubers. We used a traditional test, two traditional tests with injection of spore suspension into the tuber and next one. Previous, please. Yeah, previous. Назад верните, потому что проскочили через один, по-моему. And infection of whole potato tubers without injection. And almost all our tested strains can infect potato tubers and tomato fruits. Next one. So in Africa, there is little land suitable for growing plants, and they must be used as efficiently as possible. Next one. That is colleagues who took part in this work. Thank you. Maybe any questions?
Thank you very much, Sergey Nikolaevich. And now, please, questions. You're welcome. Have you some, please? No, when you're thinking, I have one question. Sergey Nikolaevich, to your opinion, if we, or maybe together with other countries, with Ugandan scientists, will supply them with sustainable varieties, to your opinion, what is the best way, cultivating organic potato or using chemicals as usual traditional technology?
Now, most of Africa grow potato in organic manner, in organic way, because they have no money to pesticides. And if they use pesticides, usually it's cheap contact pesticides such as mancozeb or chlorothalonil. They are not so effective and good for wet climate of Uganda. And the main problem is late blight, of course, phytophthorosis, but the next big problem is Ralstonia solanacearum. That is a soil bacteria that grow on roots inside the stem, and it is impossible to work with this bacteria with pesticides. Yes, with pesticides, they use only growth regulators. So the resistant varieties can be very good for this, to solve this problem. And if we can help African colleagues and growers to obtain these resistant varieties, that will be a very good thing for African agriculture.
Thank you very much. Other questions, please? Yes, please.
Thank you, and Sergey Nikolaevich, thank you for your report. That was really interesting. And what do you think is spectral remote sensing is suitable for Uganda agricultural technique?
No, it's an interesting question. I think that in Uganda we have two types of companies. Most of all is very small plots, fields less than 1 hectare. So it's possible to use monitoring only to understand what is growing and what yield can be expected this year. Yes, but another part of companies is a big holdings with big fields, with intensive producing of cultures, and of course, all modern technology can be implemented in this type of producers and companies.
Thank you.
Other questions, please?
Thank you. Thank you, sir, for the wonderful report. I want to ask a follow-up question to the question that Dr. Pakino asked, where she actually suggested that helping Africa by giving them resistant cultivars, and that's how you answered. But there's the issue of some pathogens like Fusarium species that are currently present in Russia, but they're not currently present in Uganda. Isn't that, in a way, in trying to help, going to actually cause more harm, where we're saying they already have a problem of Fusarium, and then there are some Fusarium species that are present in Russia, but have not been identified in Uganda?
What is the question? I don't understand. About the differences in composition of species in Russia and Uganda? What do you mean?
My question was, there was a slide that you had that had some Fusarium species that are currently present in Russia, but are absent in Uganda. There were quite a few. So my question to you is, the resistant species that we're going to give them, isn't that in a way going to actually cause more harm, because we'll be giving them potato, I don't know, cultivars or species or seed that is actually infested with species that they don't actually have, whilst they're actually trying to control the current problem that they have?
Resistant means healthy, first of all.
No, I can say that for potato, almost everywhere in the world, potato has the same pathogens. So I think when later we extract more strains of Fusarium and estimate them, I think we will find another species complexes like we do in Russia. But some group of pathogens, especially bacterial pathogens, that cannot survive winter in Russia and frozen soil. And first of all is Ralstonia solanacearum. That is the largest problem everywhere in tropical countries, in Indonesia, in Uganda, and everywhere, in South America also. So we have now this problem in Russia because Ralstonia is not overwintering here, but it can well overwinter in a wet and warm climate. And so we need to do resistant varieties also for these specific pathogens for Africa especially. It is not problem here, but it is a big problem in tropical countries. I ask you, answer your question. Or...
Yes, sir. Yes, sir, you did. Thank you so much.
Thank you.
Other questions? Please, you're welcome. In the bottom.
Big button.
Okay, thank you for your valuable information. Here I have a little doubt for you saying in Africa there is a little land for cultivation. Is it the little plots that you have displayed? Is it because of little area of cultivation or some other economic problems? In my case, I don't think there is little land for cultivation in Africa. There is plenty of bare land that need to be cultivated. So how can you justify this? Thank you.
Вы сказали, что много земли, мало земли в Африке. Это потому, что в Африке много земли. Это плодородная мало или, может, не хотелось вот в чем вопрос. Вы сказали, мало земли. Что значит мало?
These maps that I show, that is a assessment of United States Department of Agriculture that made assessment for different countries for fields and lands suitable for plant growing. So many lands in Africa are inside the desert, so they cannot be used in agriculture. But now we can say that it can be used using special technologies of irrigation, special technologies of supplying is with fertilizers and other technologies. So we can find technologies to use lands that are not included in this map, but possible for use. But the main problem will be the profit for this business. Maybe it will be very expensive, and because of this, it will not be in use. Or we can adapt cultivars of plants to grow on such lands, not without good fertility lands, неплодородные, как сказать, low fertility lands, yes, poor lands, yes, poor lands, absence of water, and other. And in tropical, especially in Democratic Republic of Congo and some other countries, big areas are under the forests, and that is not an agricultural lands also. So good lands where we can do profitable plant growing is not much.
Thank you.
And besides, I suppose I want to add a few words. I suppose that from report of Sergei Yelansky, we understand that those small amount of land, which are with high fertility, potential high fertility, they are not used in a proper way. It is necessary to improve fertility of the soil, to improve varieties, and in this case, it will be profitable, in many times much more profitable to use this small amount of lands. That is he's speaking about economic efficiency. Other questions? I suppose, please.
Thanks for the presentation. And my question is, as you said, it's the technology expensive to use in the land in Uganda or somewhere else. I want to ask, is profitable to use technology or chemical materials in the land to spend a lot of money, and at the end, you get what you need?
So you mean, can we use technology for profitable business?
Yeah, exactly. To spend money to use the technology to get at the end like good healthy fruits, it's profitable or not?
It depends on the fields. If it is a small fields, about half hectare with bad soil, who belongs to one farmer to owner, yes, and on the slope, for example, maybe it will not be profitable business. Because here in Russia, we know that profitable business for potato starts from 50 hectares. You cannot have enough money from less field. Yeah, I think in Africa is the same situation. So we need good field with a good square, and in this field we can have a good profit using modern technologies. But for small kitchen gardens, it is maybe that is profitable if the farmer makes everything his own by hands, without technique, but without pesticides. He can't obtain good yield because of late blight, for example. Late blight eat everything without the fungicides in Uganda.
Thanks, sir.
Another question, please.
First of all, thank you for your detailed information. My question is regarding the production. Of course, as you said, it's very large in Russia as well as Europe, as comparing Africa. But did you consider also the quality aspect with the application of different chemicals and others? There will be some effect. So did you compare also regarding the quality aspect, comparing among all?
You mean the quality aspects for production?
Yes.
Modern technologies, using modern technologies, we can obtain a suitable quality for food production. Of course, the quality must be estimated before you will you pass it to retailers, companies, or to shops. You must estimate the quantity of concentration of pesticides in these tubers or fruits and other components. Or if you use organic technology, also you can test the amount and concentration of fungal toxins, for example, because that is the largest problem in organic is fungal toxins. So if you use good technologies and do it correct, I don't think that will be problems with quality of foods.
Thank you.
No, I suppose enough for questions. We have a lot of other reporters. Thank you very much, Sergey Nikolaevich. And now you are moderating.
The next report, just a moment. Ekaterina Skolotneva is here.
Yeah.
Okay.
Good morning, everyone.
I can introduce the Ekaterina Skolotneva is from Federal Research Center, Institute of Cytology and Genetics of Siberian branch of Russian Academy of Sciences. And the speech, current tracking of stem rust race Uganda 99 on wheat worldwide, including Russia. So your report, please.
Yeah, thank you. Okay. One moment. So, can I speak? Yeah. My question is, can you hear me? Can you see my presentation?
Yes, we see you and we hear you and we see a good presentation. So you can
Great.
start.
So, good morning, everyone. And I welcome you to focus on the current tracking of stem rust race Uganda 99 on wheat. Stem rust is an occasional but devastating disease of wheat. Yield reduction is attributed to failed transpiration, plant lodging, shriveled grains, yield losses in outbreaks can exceed 60–70%. Farmers have harvesting dust instead of crops when level of disease is red. Stem rust is one of the most economically important plant pathogens affecting global food security. It has a wide geographical distribution and can destroy a wheat field during 3 weeks. In world wheat production, the outbreaks of foliar diseases are controlled by fungicide application and genetic protection. But fungicides have only a limited effect against stem rust. Resistant varieties provide the best protection against stem rust. Wheat is a polyploid. The complex genome carries lots of resistant genes against fungi and other pathogens. Resistant genes against stem rust we call SR genes. Most of resistant SR genes come from wild relatives as translocation into new varieties. But a new virulent gene of pathogen can work against resistant gene of wheat. Stem rust produces billions of spores. Mutants appear occasionally among them. Mutants or new races are able to attack resistant varieties. It's important to monitor the pathogen population. In the last century, a new highly virulent race of stem rust was detected in Uganda in 1999. The race UG99 arose by a somatic hybridization and nuclear exchange. This model was proposed later with evidence based on genomics. The UG99 race can infect plants in any phase of vegetation, leading to the death of susceptible plants, even with routine fungicide treatment. The question is, which plant is susceptible? The race UG99 overcame the protection coming from resistant gene SR31, which had worked more than 70 years, keeping wheat around the world free of stem rust. The SR31 gene is a translocation on wheat chromosome, which was introduced from rye. Wheat varieties carrying the SR31 gene also have broad agronomic adaptability as well as resistance to stem rust. The gene SR31 has been widely deployed in Europe, China, and the United States, and unfortunately, this contributed to the development of the pandemic situation on wheat. The statistic indicates that annual loss will amount to over $3 billion, and the farmers in Africa will suffer the most. Up to now, 15 variants have been identified within the UG99 lineage, also virulent to SR24, 24, SR36, TMP, and so on. The UG99 lineage is detected in Uganda, Kenya, Ethiopia, Eritrea, Egypt, Tanzania, Mozambique, Zimbabwe, South Africa. These were 2 waves of spores spreading. The first wave was in the northwest direction from 1998-1999, and the second to the south of Africa from 2009-2010. In June 2007, Cyclone Gonu hit the Arabian Peninsula, and UG99 appeared in the Near East, Central Asian countries, South Asia, East Asia. India is the world's second largest wheat producer. In 2021, it produced 109 million tons of wheat, 14% of global production. Pakistan is in the 10 top largest wheat countries and produced 27 million tons in 2021. India, Pakistan, Nepal, Afghanistan, Bangladesh together account for 18-20% of global wheat production. At the time when the threat from UG99 becomes clear, the majority of wheat varieties grown in India relies on 31 and 24, both of which are ineffective against some strains of the UG99 lineage. With long-distance travel of rust spores, it's only a matter of time until UG99 reaches Eastern Europe and possibly Russia. In 2013-2014, in Volga region in Russia, a race virulent to SR31 was identified among spores from cultivar Prokhorovka, spring common wheat. Next years, no more virulence to SR31 was found in the region. The tests for molecular marker to SR31 in susceptible plants were negative. Up to now, there is no official confirmation of the presence of African spores in Volga region. In Western Siberia, spring common wheat is the leading grain crop, occupying quite large area of about 7 million hectares. Here, stem rust on wheat occurs when warm, humid conditions in the early summer encourage disease development. Almost every year, across the sowing areas of Western Siberia, we plant the trap nursery, International Stem Rust Trap Nursery for UG99. This target trap nursery includes key diagnostic lines with single genes. The Siberian population of stem rust consists of local races, not UG99 yet. But again, it's only a matter of time until UG99 reaches Russia, when the weather is welcome for disease development. Unfortunately, the genetics of the Russian germplasm is limited to several genes, with 31 and 36 predominating. Currently, the researchers are testing SR14 and SR44, which they think have resistance to stem rust UG99. But up to now, more than 80% of modern varieties of wheat grown worldwide are still susceptible to the UG99 race lineage. Approaches to trace the African races are urgently needed. Molecular markers for the UG99 race lineage are still under development and validation before they can be used worldwide to prevent outbreaks. The Borlaug Global Rust Initiatives, BGRI, together with other partners of International Maize and Wind Environment Improvement Center, CIMIT, developed the primary web portal Rust Tracker for global serial rust surveillance and monitoring information. Here, one can find field surveys with incidence and severity of disease. Most countries of wheat production, about 37 at present, provide specific rust information to mitigate the threat of wheat rust diseases. Once validated and approved, data is published and automatically disseminated via a series of graphical and mapping tools. The use of fungicide to control wheat stem rust specifically related to UG99 race lineage has very little published information. Moreover, this option is too expensive for many small farmers in the developing world, and they rely on gene resistance to control wheat diseases. We believe that an integrated approach using resistant genotypes, together with application fungicides and biostimulants, we tested now, testing will take a step forward in protecting wheat crops from stem rust. Thank you for attention.
Thank you. Maybe any questions? So I can ask one question about Uganda. Is this race of rust is harmful for wheat in Central Africa, or it goes out from Central Africa and it give loss to other countries, but not in Central Africa?
No, of course, it appeared as very harmful agent for local wheat cultivars in Africa for first time, and as I said, these races arose by somatic hybridization, but on wild relatives, and then they jumped to releasing cultivars, and more than 80% of crops was damaged at that time. So first of all, this race is harmful for African crops of it.
Okay, thank you. Any other questions?
I have one. And please, is this race of rust dangerous only for wheat, or it can affect other cereal crops?
Okay, thank you for question. Yes, this race is also can also damage barley. And barley is the second cereal which is cultivated in countries. Yes, so it's a very important crop, and UG99 can infect barley as well.
Thank you.
Other questions? No? You're welcome.
Thank you for the presentation. I just have one question. You mentioned that currently it hasn't been detected in Russia. What are the measures that have been put in place or can be put in place to prevent the spread of UG99 in Russia or in other countries?
Some preventive measures, yes, you ask.
Yes.
Okay, yeah. Along the way of UG99 spreading, now breeders plant the resistant cultivars with other genes. SR25, sorry. I said about SR14 and SR44. Yeah. So the first step is using resistant cultivar. And the previous step of the first step is checking, is analysis of genetic, of genetic analysis them with varieties which we use. And this analysis, most of countries have performed this analysis, and we know this information, and we know that unfortunately 80% of varieties all over the world are susceptible. So now we see the work of shifting the releasing cultivars, especially along the way of UG99 spreading. It's very popular to use these gene pyramiding technique, when in one genotype we accumulate more than one resistant gene, not only SR31, but also SR2, which protect the plant during the adult phase of it, and so on. So another the next measure is to monitor the pathogen population, and here also two ways how to do it. First of all, researchers in each country plant special trap nursery I showed you, and this trap nursery consists of wheat lines. These wheat lines are susceptible to UG99, and we see infection. That means UG99 is here. So this measure is a visualized measure, right? Other step is analyze spore collection to collect spores in field and analyze them in the laboratory. We do it here in Siberia. Also, scientists collect spores and send them to Denmark. There is a hub to stem spores. They collect spores from each from many geographical location and analyze them for UG99 presence and inform breeders. It's very important that this hub located in Denmark, like other Russian hub in Siberia, here the weather is not favorable to development, especially UG99. So we can study here UG99 carefully, and we are not afraid of spreading it out. What else? And of course, I said about application of fungicide together with biostimulants. Now we study this topic when biostimulants can push special resistant gene to work, push the immunity of plant against stem rust. Yeah.
Thank you. Thank you. You really answered in detail. Thank you so much. Thank you.
Thank you for question.
One more question. Welcome. Yeah, you are welcome.
So thank you for your great presentation. Okay. Thank you for a great presentation. I would like to ask you about this, the level or the extent of destruction that has already occurred by this stem rust. And not only that one, as we just know that this wheat is a leading crop, a leading staple crop. And as you have indicated, almost 80% of the varieties are susceptible to the stem rust virus. So this indicates that this is a great threat for the global food security program. So do you have any intention of just introducing several resistance genes to the wheat varieties so that we can upgrade the process of resistance in the plants? Because if we just introduce only one resistant gene, that may be a problem, because the pests or the disease itself will just manage itself so that it can resist the process of the plant's resistance. So if that is the case, I think it would be good to introduce several genes so that it will take time for the pathogen to resist or to just, to tackle the crop. So do you have any intention of introducing many genes? And the other one is, what is the level of damage that has already occurred on the plants? Can we just quantify it on percentages on a global basis? To what extent or to what percentage that the wheat production has already been harmed as compared to other diseases?
Okay. What you said, you just emphasized the main idea of my report, yeah? And as I say, gene pyramiding is one of the measures to prevent this catastrophe, right? To obtain and plant genotype with pyramid of genes is the best decision. And now researchers test a lot of combination of genes. For example, SR25 plus SR31, SR2 plus, and so on. Yeah, this work is going now. First, right, and as when I say 80% of varieties, it doesn't mean I estimated diversity of varieties, global varieties, and not those variety which is in majority. If I understand your question, your first question. And
yeah, actually, the first question is, you're just answering it. What I mean is, have you been trying this to introgress or to transfer several genes at a time? I don't mean individually.
Yes,
several genes.
At the time, not only now, already more than 10 years, breeder have been planting these pyramiding genotypes. Yeah, of course. Because, now it's 2023 and UG99 was appeared in 1999. So, and then next about numbers you asked. Unfortunately, it wasn't clear for me. Your second question.
Actually, the second question is related to the percentage of damage that is accounted for stem rust damage only as compared to other diseases. What percentage of damage has this stem rust contributed to the global destruction of this plant?
Okay, I'll show you. My presentation. Just a second. Okay. I'll try to find. Okay. Закрыто. You ask about African continent or global food security or what? Okay. Every year percent is changeable. For the first years, they wrote about 80% of damaged crops in African continents. And every place the UG99 come, they reported a high percent of damage. And usually stem rust is problem for wheat. Local stem rust races is very high problem for wheat in every wheat countries, but usually breeder plant resistant varieties to local stem rust. And the problem was these African spores. That's why the high percent, around 80% of damaged crops they had. I tried to find some illustration with numbers, because the numbers is very.
I don't mean about numbers. I just want to know only about its extent, the extent that it caused the damage, as compared maybe if it is very harmful, it may just attract our attention to just deal with it. So that's what all I want. I'm not asking you about numbers. Thank you so much. Thank you.
Окей, но мне нужна, наверное, помощь. Я не очень поняла, что задается, о чем мне нужно рассказать.
Ну, предлагается просто crop rotation как метод борьбы. Я полагаю, что вы уже ответили на вопрос.
Ну вот мне тоже кажется. Sorry.
Коллеги, I'm sorry, we are out of timetable, so we must proceed with our reports. It was very interesting report of Ekaterina, but we have no time for further discussion of Ugandan race. I'm very sorry. So I want to introduce our next speaker, Krutikov or Terехov, who will have a speech.
Hello?
Alexey Terехov, yes?
Yes.
Alexey Terехov is head of Khabaragi Company Limited, and he will introduce us a report of on the item Stabilized Colloidal Silver in Agriculture, a new approach to crop protection and plant growth stimulations. So you are welcome.
Ну все, наверное. Ой.
Okay, one second. Видно презентацию, да, Сергей Николаевич?
Yes, we can see. Very good.
Okay.
And hear you.
Okay, so dear colleagues, thank you for the opportunity to present a range of colloidal silver-based plant protection and plant nutrition products developed by the scientists of the Lomonosov Moscow State University to you today. So why colloidal silver as an active ingredient for plant protection products? Well, first of all, because it allows you to achieve several positive effects, all while maintaining eco-friendly nature of your farming. So these positive effects are plant growth stimulation, which includes the improvement of product quality and the increase in productivity. This effect is obtained by affecting the hormone system of a plant by reducing plant sensitivity to ethylene, the hormone of aging, and by increasing the amount of auxin, the hormone of growth. Then there is the immune system boost, which allows the farmer to protect their plants without involving traditional pesticides. Then there is stress tolerance improvement, the improvement to factors such as drought or high temperature, low temperature, ultraviolet radiation, all these factors, I'm sure that African farmers are dealing with on a daily basis. And finally, there is crop protection. Colloidal silver products allow you to protect your plants not only from fungi, but also from bacteria, which is something that is very hard to achieve unless we're dealing with antibiotics. And these products are not antibiotics. They are completely eco-friendly. They leave no residue in plants, which is something that we had to prove time and time again while registering our products around the world. As I've already said, these products were developed by the Lomonosov Moscow State University, and our team was the first team to register colloidal silver activated by using biologically active stabilizers as an active ingredient for plant protection products almost 10 years ago. And currently, these products are patented in more than 20 countries, countries such as the United States, the European Union countries, China, Japan, South Korea, Chile, and so on. And these products are promoted in 35 countries around the world. They can be easily bought by a farmer and used either to stimulate growth or to protect their harvest from different types of pathogens in countries as diverse as South Korea and Turkey, China and Uzbekistan, Brazil, Chile, Ecuador, Panama, Honduras. So more than 15 countries. And as you can see from the map, the products were also tested in 4 African countries. These countries being Uganda, the Gambia, where colloidal silver was applied to rice. Rice is one of the main agricultural crops that are very much affected by colloidal silver in a good way. Also, they were tested in Angola, where colloidal silver was used together with a traditional pesticide in order to lower the amount of pesticide needed. This mixture of a traditional pesticide and colloidal silver products was used to treat tubers of potato. And finally, just last month, we obtained results from Mozambique, where the products were applied to tomato, and the farmer saw that the harvest was much bigger compared to untreated plants, and the quality of tomato itself, the fruits, was also better. So we see a lot of potential in terms of application of colloidal silver in African countries. First of all, because these products work a lot better in countries with hotter climate. So that's why some of our best results are obtained in tropical countries or countries with desert-like climate, or countries where there's hot temperatures. Some of the time, countries like Uzbekistan. Then there is the question of little land that can be cultivated, as was already mentioned by several speakers before me. And with the help of colloidal silver products, you can make your farming efficient, even if there is little land to be cultivated, just based on something that we notice in South Korea, where there's a lot less land that can be cultivated, yet the results show that by using these products, the farmers can still get great harvest. And finally, there is the fact that some of our best, most remarkable results that we have obtained over the years. They come from using these products with regards to crops that are very popular in Africa. So I would like to show you some slides, and if you have any questions or comments, I will be happy to answer them. But in order to save time, I will just show you those slides, and if you have any questions, please let me know. So these are the results that we obtained while using colloidal silver products when growing wheat. These are the Russian results. These are results from China, where colloidal silver products were applied to Areca catechu, which is a palm tree that grows in Southeast Asia. These are results we obtained in Turkey when using the products when growing potato. These are results that we obtained in Chile while treating different kinds of nuts, so almond and walnut. These are results that we obtained in Peru when treating Peruvian lime. As you can see, the increase was almost 20%, which is rather remarkable. These are results from Ecuador when corn was treated, the 22% increase. These are the results also from Ecuador, which we obtained by treating rice. As you can see, the increase was extremely high, 66%, which is not something that we often see, of course, but I would like once again to stress that rice is one of the main crops affected by colloidal silver. These are tomato results, 37% increase. And this is the cocoa results. Our products are often used in order to treat cocoa and coffee. These are results from Ecuador, where our goal was to inhibit and eliminate phytopathogens. As you can see, the difference is like night and day. And finally, these are results from Nicaragua where the products were applied to tobacco. So thank you very much for your attention, and we welcome all kinds of collaboration, and we would be happy to help you bring these new products. Once again, these are new products. They're not generics. They're not produced anywhere in the world except Russia. So if you have any questions, I will be happy to answer them. Thank you.
Thank you. Any questions? Welcome.
Thank you very much for the presentation, and I have just two interesting questions, especially from the countries which you already did trials in details with this growth stimulator. Do you think there are any, I would say, future effects? And what makes your product so unique? Exactly. Thank you.
Okay. So future effects, you mean possible negative effects that can occur in the future?
Exactly, sir.
Okay. So so far, even though these products have been in use for more than 10 years already, we haven't, first of all, we haven't seen any signs of resistance, which is something that is extremely serious in terms of chemical pesticides. So if you, for example, Turkey is one of our main markets, and the situation is very bad there because farmers do not follow the rules. They simply use the same products year in, year out. So very soon pathogens become resistant towards traditional pesticides. But so far, we haven't seen any kind of resistance to colloidal silver. And at the same time, there is little accumulation of silver in the land. So even if you are using it for 30 years, let's imagine that, the amount of silver accumulated in the soil would that comes from these products would be much less than what is naturally present in the soil. So once again, there is no residue. So if you spray your plants with these products, those are liquid products. So if you spray your plants, it doesn't enter the tissue of the plant. It stays on the surface, and then when you simply water your plant, the products end up in the soil and they are deactivated by elements present in the soil. So far we haven't seen any negative effects, and of course is something that we have to monitor, and we do that, but so far we haven't seen any negative results. And as for your second question, it's the fact that it's basically 3-in-1 kind of product. So first of all, there's growth stimulation. There are very many biostimulants, fertilizers on the market, yet a lot of, in fact, our team was the first team in Russia to talk about elicitor qualities. And now, if you check Russian market, there are many products, they say that they also have elicitor qualities. Elicitor qualities is basically something that makes your product work as a vaccine. So the plant sees it as something that is not natural to it, even though colloidal silver is completely safe. But as a vaccine, it makes the plant boost its own immune system, grow quicker, grow bigger, and this helps you basically protect it even when you don't use chemical pesticides. And I know that getting rid of chemical pesticides to a possible extent is one of the goals that African governments are trying to achieve these days. And also, it's the fact that there are little to none products on the market that can help you fight bacteria. And Professor Yelansky was actually one of the first people noticing the positive effects of colloidal silver in terms of bacteria. I'm not sure about African countries, I have to admit, but most countries do not allow the use of antibiotics in agriculture. So if you're dealing with fire blight or late blight or different types of bacteriosis, the only choice you have is to chop down your olive tree garden, your apple tree garden, in order to stop the bacteria from spreading. However, with these products, you can easily do that, and there are no other choices that you have unless you're ready to use antibiotics. But most countries, once again, they do not allow to use antibiotics. Some do, like the United States, Chile, but for example, in Chile, they use our products for growth stimulation. So you can pick and choose whatever you want. For example, I know that we are running out of time. In Russia, last year, most farmers decided to use our products instead of chemical pesticides. They did not see any growth, so the results they obtained were the same. However, it helped them cut down on their costs Because if you do not see any growth, but instead of expensive pesticides, you use colloidal silver products, which are less expensive, you get the same results. You maintain the same level of quality. This is still good. You can pick and choose whatever function. They are multifunctional, as I've already said, and they also completely safe. You don't have to think of the consequences of using these products. Okay, thank you.
Thank you.
Okay.
Only one very short question because we are out of graphic.
All right. Good morning, and thank you very much. Just wanted to ask if there is a relationship between the colloidal particles and nutrient use efficiency. These are soil colloidal particles, isn't it?
Obviously colloidals. I mean, yeah, colloidal particles are bigger than nanoparticles. Yeah, but the products themselves are liquid, so they are like 99% water. Yes.
Yeah. So do they improve nutrient use efficiency when we use them in our soils?
They affect the plant in a way that they become more receptive towards micro and macronutrient intake. So there is this system of delivery. These colloidal particles, they act like little ships that carry those micro and macronutrients, and so it goes to the cell much quicker, and the cell intakes whatever nutrients are needed. Yeah.
Thank you very much.
Okay. Thank you.
Okay, so we have very small time because we are out of graphic, and we have 15 minutes and 5 speakers. So I'm sorry, and we stop this discussion, and I want to give a word to the next speaker, Joseph Nyamboze from Zambia. Joseph, are you with us?
Joseph, good day. Can you answer?
No, if there is a problem with Joseph, so the next speaker is Guruleva Anastasia from Moscow Technical University, named after Bauman. Spectral methods for monitoring the state of vegetation.
I'm here. Presentation number 5, please. Включите, пожалуйста, презентацию номер 5 для Гурулевой. Спасибо. Hello, dear forum participants. My name is Anastasia Guruleva. I am a senior lecturer in Bauman Moscow State Technical University and a researcher in Scientific and Technological Center of Russian Academy of Science. So I am an engineer, and me and my colleagues are developing imaging spectrometers. And also our group is engaged in their application in agricultural technologies, and the aim of my report will be to discuss it. So please, next slide. Let me quickly remind you what imaging spectrometers are. Please, next slide. Thank you. You can see the typical plant reflectance spectrum, and you can get it if you will use conventional spectrometers and acquire by it irradiation that reflected from plants. Please, next slide. And now let's talk about imaging modalities. If you have a monochrome camera, you can obtain monochrome images of your plant. And if you want to, if you use RGB camera like in our cell phones, you can obtain images that includes three other images, red, green, and blue, and you can get it by optical filters. And next slide, please. And the next step is imaging spectroscopy. If you will use not only three filters, but more, 4 and up to 25, you will use multispectral imaging. And if you have more than 25, as a rule, is about 100 filters and 100 spectral images, it is hyperspectral imaging. That's pretty easy. And the key features that you can obtain, curve like you see at the top of the slide for each pixel. That means that you can map parameters of your objects, for example, plant. Please, next slide. In our center, we develop both multispectral and hyperspectral imaging spectrometers. Their photo you can see. And hyperspectral has better spectral resolution. The multispectral is a small, have a small weight and size, so it's compatible with the drones, and you can have high performance with them. In this report, we will focus on multispectral, invisible, and near-infrared spectral range, hyperspectral images in visible and near-infrared, and infrared devices. Next slide, please. And now let's talk about application. And next slide, please. The first and most urgent application is a chlorophyll content estimation. Please, next slide. Traditional methods are very labor intensive and time consuming, and most of the a lot of the study is dedicated to find the method that will replace them. And spectral imaging seems to be very promising for this goal. Next slide, please. Next, пожалуйста, slide. Okay, thank you. Laboratory methods have the best first spatial and also spectral resolution, but respectively low performance. Next slide, please. The ground-based devices is a medium in terms of performance and spatial resolution and spectral resolution too. And next slide. And the high performance typical for remote sensing. This method is implemented as a rule with multispectral sensors devices, and selection of band for these devices is a key factor in terms of the chlorophyll content estimation accuracy. So on the one hand, we want to replace traditional analysis, and on the other hand, we want to choose the spectral band for current application to design multispectral devices. Next slide, please. So to implement it, we've developed the following approach. that will be shown on the next slide. First, we collect the plant leaves with the large chlorophyll content range. We can implement it with one crop in different development stage, or with multiple crop. Then we measure reference data and choose the suite of the vegetation indices and acquire spectral images right in wavelength that they need. So we collect hyperspectral data too. When we compare this data, reference and hyperspectral, find the model, and after that we can map chlorophyll content, as I show you later. Next slide, please. And next slide to be more briefly. So you can see the experimental setup. It includes hyperspectral imaging, imager, and we acquire images only for plant leaves. As you can see, next slide, please. And next slide. This is a sample of the images, spectral images, and also we developed image processing algorithm. I won't go in details, but it's really important. Without it, nothing will work. But next slide. Referent measurements referent measurements were implemented using traditional spectrophotomic analysis. The protocol will show on next slide. And next we calculate index map. You see RGB image, and next index map, and after that we estimate the most accuracy index and calculate chlorophyll map for this index. Please, next slide. Yeah, this is a model. A model for index and chlorophyll reference chlorophyll concentration. So we use this model to map chlorophyll content. Next slide, please. Yeah, these two type of maps at the left side of this slide. This maps for leaves at different development stage, and on the right side for different crops. Next slide, please. Okay, and so now we have method that can replace traditional method because it has really good accuracy, relative error less than 10%. And also we have multi-crop model, and more we choose a spectral band to design multispectral camera. Next slide, please. Another application, briefly, next slide please. It is an impurity assessment. There are standard methods and there are spectral high performance and automatics ones. Yes, next slide, please. Hyperspectral method described in many studies, and our novel contribution is method to assessing impurity in. Oh, next slide. Next slide. Next. Yeah. To assessing impurity in a heap of grain. So we, with the hyperspectral imaging, we assess samples with different impurity, develop a neural network model. Next slide, please. Classify impurity and obtain classification model accuracy at more than 93%. And next slide. This is classifying. Yellow is impurity seeds, and this is our classification model accuracy, more than 93. Next slide, please. Final application, food quality analysis. Next slide, please. Hyperspectral imaging is also very promising for early detection of the defects. For example, you can see fungal contamination during decay. We examined peaches, next slide please, with various defects on our 3 instruments and obtained typical spectra. And the next step is the defect imaging and the criteria of defect differentiation. Next slide, please. And you can see our colleagues we work with, and Rudan employees among them. And we all believe that hyperspectral imaging is a really powerful tool for monitoring tasks and in agricultural technologies too. So we still have a lot of interesting work to do. Thank you for your attention.
Thank you. Any questions?
I have a question. How this equipment can be moved in the field? Because we must make monitoring of big amount of square meters, and how it can be moved?
Multispectral camera can mount on the drone. Hyperspectral imager could be in handheld devices. So that's real.
Thank you.
Very short question, one more.
I just wanted to ask how we can apply this technology in weed management and disease monitoring.
Disease monitoring, this is one of our area of interests, and we have some study for it, and for food quality control and for plant health. So I can discuss it in detail if you want later with you.
Right, thank you. That would be interesting.
Thank you. So you want to talk?
Yeah.
Thank
you so much for your presentation. Since, of course, we are here to study, to take back to our countries in Africa. And I believe this forum is an eye-opener for us. I want to know, of course, they've asked one part of the question, that is, this instrument can be mounted on drones to take to the field. And how will it be possible for students to learn from your technology?
Students, first of all, you can, and Rudan students already do that. You can visit our building, and we can show you how it works. And also we trying to design more cheap device that could be used with with students and other. Okay.
Thank you for interesting presentation. My question is, what's the main target for building the model and using the spectroscopy?
Oh, could you repeat, please? I can't understand.
What's the main target or the main point? What's the purpose of building the model?
The main point is to define the most accurate index, and after that determine the coefficients in model to that allow to recalculate index to chlorophyll concentration, because as we because for farmers, vegetation index is nothing. They maybe know something about chlorophyll concentration, but index isn't really suitable for them. So we need to recalculate. And for accurate recalculate, you need model.
Thanks.
Thank you. So now I see Joseph Nyamboze connected to our meeting. So I pass the word to Joseph Nyamboze, PhD student of the Agrarian Technological Institute, RUDN. Russia, Africa, ties in agricultural technologies, the use of smart agriculture and precision farming as a way of addressing global climate change and hunger problems. Joseph, you are welcome.
Joseph, we do not hear you. Please check your microphone. It is switched or not?
We can't hear you, Joseph.
And now we see there is no connection. Joseph, now try to speak.
Hello, hello.
Oh, now we hear you.
Okay, all right. So, dear colleagues, today I'm going to present on a title, Russia-Africa Ties in Agriculture Technologies, the use of smart agriculture and precision farming as a way of addressing global climate change and hunger problems. Firstly, I would like to give a little bit of the background since we are talking about the ties between Africa and Russia. I want to mention that the cooperation between countries plays an important role in shaping the progressive economic systems, and Russia and Africa, in particular Zambia, because I belong to Zambia, are no exception. And historically, Russia and Africa have had close ties, and these ties have been in the education sector, and as well, Russian universities, they have been offering scholarships to African students, which has facilitated their studies in Russia. And this has created a long-term cooperation and has expanded education opportunities for African youths. Now, I believe if we take the implementation of joint research in fields like agriculture, it is as well a promising area of cooperation. Joining efforts in fields of scientific research will make it possible to obtain new knowledge and share innovations which will benefit all our countries in partnership, socially and economically. Now, since today I'm trying to address on a topic to do with ties, the question that I would throw is, why promote ties in agrotechnologies? Why use smart agriculture and precision farming between Russia and Africa, or in particular Zambia? Well, I would say it is now, looking at the times that we're in, is one of the effective ways that we improve the soil fertility. I heard some other speakers that were talking about the impoverishment of the soil statuses in Africa. I believe by these ties, we may as well look into other aspects in which we can improve the soil fertility. Apart from that, the increase in crop productivity and as well the surety way to address the climate change and hunger problems. For example, literature or studies, they are able to give, they are able to reveal this, in particular the Western African countries, as being vulnerable to climate change and the variability on the account of social and physical characteristics. Well, in this area, the western part of Africa, farmers are subjected. They have to cope to highly variable, short, and predictable rainfalls. And yet, in this area, these people, they just depend on rainfall for them to be able to do like their agricultural practices. And then you see that there is an increasing variability in climate change, and water resources in these areas, they become more unpredictable. And we understand very well where there is no water, agricultural practices becomes a problem. Well, apart from that, in addition, I would like to say that increased runoff frequency has as well contributed to soil erosion, especially in most African countries. Now, maybe my experience, I've been in Russia for quite some time, but my experience, the African experience, you'll find that in Africa we have torrential type of rainfall. The rains are very stronger as compared to Russia. So that has as well contributed to the soil erosion. Soil was being eroded. So I believe these ties in which we can bring in technologies that will be able uh, to moderate or maybe allow us to be able to improve our agricultural lands, then we'll be able to realize the production to increase crop productivity. Then I would want to say as well, according to the studies that have been done, agriculture sector contributes approximately 10 to 20% of the total anthropogenic greenhouse gases. This is in line with the global warming that we're talking about, the climate change that we're talking about, which is the topic of discussion. And we understand very well that as well studies do reveal that this climate change has got a negative effect on crop yields, livestock production, thus threatening food security, especially in vulnerable continent like Africa. So therefore, when we are talking about ties with maybe countries that have advanced in technology like Russia, and they get to unite with the African countries, I believe that problems to do with the global warming and the climate change, we may get a long-term solution to these things. According to what was presented to do with the technologies with the United Nations, the Department of Economic and Social, they had to discuss interesting message that to me I found it interesting, especially that we are talking about this topic. And the key points they highlighted are these that are presented here, talking about the accelerated application of smart, digital, and precision agriculture technologies that offers a historic opportunity to improve agricultural productivity of smallholder farmers and developing countries, and to generate the rural transformation needed uh, for achieving the sustainable development goals by 2030. We are not yet in 2030. So I believe these points, they really make sense for all of us. Many of the challenges that are faced by smallholder farmers are as the result of the information asymmetries and deficiencies, which I heard one of the speakers talked about educating the farmers in Africa, lack of information that is there, which I totally agree. There's a lot of asymmetries and deficiencies in terms of information. Therefore, the frontier technologies can mitigate these challenges by providing farmers with real-time precise data, analyzes and that inform their decision making at every stage of agriculture cycle. Despite these benefits, that's the point that I want to highlight here, the uptake of technologies by smallholder farmers remains limited. To address this challenge, the adoption of these technologies, it is important to invest in digital literacy in rural areas, establishing new generations of agriculture extension, smallholders, and build up infrastructure for agriculture economic commerce. So I believe this information is very important, especially for development countries. In this area, I would like to highlight, as a PhD student, uh, researching on the use of biological preparations and improving the nitrogen use efficiency by plants, in particular wheat, spring wheat. As an alternative to increasing crop yield and quality, I'm optimistic that ties between Russia and Africa in areas of agrochemicals and biological preparations will have a positive impact on crop production and quality assurance in Africa, reduce hunger problems in certain parts of Africa, and the world at large, not just in Africa, but as well and the world at large. Now, aside the biological preparations that I'm researching on as a PhD student, I'm as well looking at, my research focuses as well on the use of stable nitrogen isotope to study the uptake and utilization of nitrogen by plants. It is a common knowledge that nitrogen fertilizer application is essential to crop yield. We know that. But it is often accompanied by nitrogen losses that are detrimental to the environment. We're talking about global warming again, climate change comes in. Apart from that, also causes economic losses to the farmers because we apply these fertilizers and we just lose them in the air, like in form of gases. So that becomes a loss for farmers as well. Currently, studies as well, they are able to reveal that less than 50% of the fertilizer applied to the cropland is converted to harvested products. And the reactive compounds, nitrogen compounds, are lost to the environment, causing serious problems, commonly known as the eutrophication. I think it's a common problem that these fertilizers have brought about, and as well even loss of biodiversity and climate systems perturbations. So in regard, the use of nitrogen 15 is to ensure that we get to track the nitrogen flows in the agro systems so that we can be able to moderate the application rates and as well to moderate the losses that we get to experience as we get to apply these chemicals, and as well even the fixation that happens in the soil. So basically we can be able to track the flow of nitrogen in agro systems. On this, I just picked this part, I got it from my dissertation, and I would like just to cement from what I've already said. These are the results that I got in 2020 that talks about the flow of nitrogen and in spring wheat, and that was in the phase of flowering phase. Now, when you look at this, I want you to observe, especially on the absorption rate of the plants from the fertilizer and the immobilization in the soil and the loss of nitrogen in the air. You realize to say on the variants where the nitrogen, where the fertilizer purely was applied, the loss is a little bit higher. You're able to see 7 there. And then regions where you have the nitrogen fertilizer and the biological preparations that were used there, you are able to see that there's a little bit of the reduction in the loss. And this is exactly where we are interested. We don't need to lose these fertilizers. Our aim is to allow the plant to maximize the usage of these nutrients, because the use of these nutrients is what will ensure quality in plants. So this is exactly what you're able to see, that the biological preparations had an effect on the reduction, on the losses of the nitrogen itself. And in areas where you have maybe the dosage 90 there, you are able to see that the loss was quite big. So basically, the biological preparations and as well even the chemical fertilizers, using them together, they are able to make some sort of regulations on how the nutrients from the fertilizer may be used. In conclusion, because of the time, I would want to say that the ties between Russia and Africa Agrotechnologies and use smart agriculture and precision farming as a way of addressing global climate change and hunger problems has a great impact and prospects which should not be underestimated. Development of joint research projects in other fields aside agrotechnologies will strengthen ties between Russia and Africa, Zambia. Thank you for your attention.
Thank you, Joseph.
If there are questions, I'm here to answer.
We have no
I
time
suppose
for
there are no
questions.
questions because the report was distinguishedly made and we saw all information on slides. And now, dear colleagues, we must interrupt our session. We must have coffee break and all other reporters will have opportunity to speak after coffee break. Please, now we must interrupt and have some rest and some meal with coffee. You are welcome.
All right, thank you so much. Thank you.
And we are waiting for you in this hall after in 15 minutes.
Oh, I'm in Zambia right now.
Joseph, we have breathe, so go drink some water, right?